There are certainly lots of options for Mendel to have what I would consider variations. So Mendel had very specific rules he laid out, and we follow those a lot, but there are certainly times when the idea that you'll have a dominant and a recessive allele and only two options, this is rare. Okay, we do see it a lot, but we certainly see the exceptions almost as frequently. So a couple of things that we need to consider. Something called incomplete dominance. This means that if you're a heterozygote, you are not going to express only this dominant trait as we would normally expect. Normally we would say you would show the dominant trait. But in incomplete dominance, you actually get a case where neither allele is dominant over the other, and you would actually see both alleles present. Okay? This is not blending. We got rid of blending lectures ago. Okay? So if you cross two flowers, one red and one white, and you get a red, a pink, and a white flower, we can look at this very differently than just saying that they're being blended together. You have to keep in mind that any color in an organism is pigment based, so it's based in protein. So what we need to think about is if you have only one allele, okay, so say with our red flower, we would see red, big R for red, big R, little r actually gives us pink, and little r gives us white. Why is big R, little r giving us pink and not red as we would predict? With one, think about this as concentration. With one big R versus two big R's, you're getting half the protein that you get with big R, big R. So pink is not so much a blending of red and white, and it is not a blending of red and white in genetics. It is half as much protein. So they show you here. We've crossed our red flower with our white flower. We get this pink hybrid that's a heterozygote. Fine. But if we would cross that heterozygote, how do we know that this pink flower is not blended? If this was blending, we would always get pink flowers from here forward. We don't. We have the same issue Mendel used to have. We're getting red again, we're getting white again, we're getting a reoccurrence of these traits. Fine, that's good, because that actually means we have a different characteristic. We have something known as incomplete dominance. We see this in disease as well. This disease is actually associated with your production of cholesterol. Okay? So when we look at this production, what we are concerned about is having high cholesterol levels. LDL is bad cholesterol. But what we look at is the ability to keep that bad cholesterol from circulating in your blood. We actually convert LDL to HDL, the good cholesterol in your body. We want HDL. So our job of our cells is to take up LDL and make it into HDL. Okay? So in the case of our normal cells, we've got lots of these LDL receptors that are going to take up that LDL for us. In a case of our disease here, we have no receptors. So we're not taking up any LDL. But in the case of this mild form of the disease in the middle with our heterozygote, we actually have a fewer number of receptors. So receptors are proteins again, right? So if we have protein that's made in less concentration, we have fewer receptors. So we have some intermediate form of this disease. Incomplete dominance. Now, we also have number of cases where we see alleles that can be expressed together. Now this is a weird one for us too, meaning that you can have a diploid individual carrying two alleles and you would actually see both alleles. Blood type is actually the kind of common example for this. So you can be blood type A, you can be blood type B, you can be blood type O, you can be blood type AB. I'm not worried about the plus minus at the moment for those of you, or positive, negative for those of you that are interested. I just want to show you the basic blood types for a minute. So what we see with this is actually something known as codominance. Neither allele is dominant over the other one. So we actually can get, if you are AB genotype, you actually express AB blood. So what this refers to is actually surface proteins. So this means that our proteins are co-expressed. You get A proteins here, B proteins here, A and B proteins here, and no proteins here. So having an O blood type means that you have no surface proteins. Do you know what kind of cell this is? Well, it tells you up here at the top. It's a red blood cell, okay? Pretty characteristic shape and size, but what we're interested in with blood type is what proteins are on the surface. If you are blood type O, you have no surface proteins at all. This is not a bad thing, okay? It just means that you don't have any proteins on the surface of your blood, of your red blood cells. So we can actually look at this formation and whether or not those are forming as they should, these protein carbohydrate groups. So for A blood type, you can actually have two choices of genotype. B, two choices. AB, you only have one choice. And O, you only have one choice. You have no proteins on the, or protein carbohydrates on the surface. So this little II is our recessive. So you can be homozygous dominant, homozygous codominant, or heterozygous and be B. You can be homozygous codominant for A, or you can be a heterozygote, okay? You see there are options here, but this is the one that's most interesting, this AB, because you can actually have what we would normally consider a heterozygote state, but because A is dominant and B is dominant, when you put them together, they're codominant, and you see both A and B. We can test for this in the lab. We actually react it with antibodies to consider whether or not the blood actually clumps, okay? And this is why when we look at individuals with particular blood types and you get typed for blood at the hospital, they want to know what your blood type is in case you need a transfusion, because they don't want to transfuse you with the wrong blood. If they do, the blood could clump and kill you. Okay, so this clumping reaction is the bad one. We don't want to see this heavy level of clumping. So for individuals that are type O, we actually see a lot of clumping if we give them A, B, or AB blood. Why is that? Well, they have no surface proteins, none of these carbohydrates on the surface of their red blood cells. So if you give them these other blood types, their body actually has a negative reaction to it. But you notice with type AB here, I can give you, if you have type AB blood, I can give you any type of blood at all, and you'll have no reaction. That's because your body is used to A carbos, B carbos, and AB carbohydrates and proteins, okay? Now O, it doesn't have the surface proteins, but your body's not concerned about that, all right? So you can see that and how this actually works out in these different scenarios, just of interest. We also have something known as polygenic inheritance. Poly means what? Many. So if poly means many, we're looking at inheriting many genes that affect one trait. I show you height here, so it might not surprise you that the girls are in white and the boys are in blue in my distribution here on the bottom left. So we look at the height distribution. If you can read the numbers on the scale, it goes from 5 feet to 6.5 feet. Okay? And on that continuum, you see a distribution of females and males. Well, what affects height? There are lots of things that affect height, right? So mom and dad, first of all, you got to get the genetics in there. But then what else? Things like nutrition. We can think about whether or not you had childhood sickness, a lot. Childhood illnesses contribute to stunted growth. Okay, if there were any other conditions that you had when you were little that might have impacted your growth patterns, can affect height. So all of these things come together to give you one trait. We'll look at this in bird beaks as well. There's a distribution, kind of a normal curve distribution here looking at beak height, but beak size is the same as height in you. It's affected by many, many things, meaning it's polygenic. Many genes, one trait. Okay. Now we can look at how I mentioned environment for you as far as nutrition and its effect on your height. But we can change things like pH. So hydrangea, some of you may know these as snowball bushes. Their flower color is actually variable based on pH. pH has a very significant effect on proteins, right? So if proteins are affected by pH, high pHs or very low pH will change that protein. If color is protein, then any change in the protein changes the color. And you can actually get them to change colors by changing the pH of the soil. Pleiotropy is one that we don't see a lot, but this is, some folks think about this as the opposite of polygenic. So pleiotropy is one gene that has multiple effects. Polygenic, remember a minute ago, was many genes, one trait. So pleiotropy is one gene, many traits. I give you a couple of examples here. Sickle cell and Fragile X. Fragile X is associated with the X chromosome. This does not mean that it only affects women. It will affect women and men. So for sickle cell, though, we actually look at the type of hemoglobin that's made in your red blood cells. And so with sickle cell anemia, we look at the buildup of the sickle cells, or the different red blood cell shape. They're kind of shaped like this instead of nice and round. So what actually affects this and what can happen because of it? Well, we look at anemia because of it. We look at damage to your organs because your red blood cells are now shaped like a sickle and they literally can cut tissues. Okay, and it's also related to your susceptibility for malaria. It turns out if you're a carrier of sickle cell, you actually have a decreased susceptibility to malaria exposure. We'll talk about this more later. Very interesting. So your normal hemoglobin, up at the top here, mutant hemoglobin changes one base pair. One single base pair in your entire DNA changes to end up with this mutant hemoglobin, which changes a single amino acid, which ends up producing the wrong protein. When we produce the wrong protein, we develop sickle cell anemia, Okay, here's your sickle cell, so you can see it in the middle. Produces this abnormal hemoglobin protein, and we end up with abnormal cell shapes, and as we mentioned before, it can have all kinds of issues associated with organ damage. It can also lead to all sorts of other effects, susceptibility to pneumonia and infections, pain, joint problems, general weakness. If your red blood cells are shaped like a sickle, remember red blood cells' job is to carry oxygen. If it can't deliver oxygen because it's the wrong shape, you end up with all kinds of associated conditions with low oxygen supply. Fragile X, on the other hand, is associated with the X chromosome, as I mentioned, and has very highly variable expression. So, like with sickle cell, this is a pleiotropic disease, meaning that you have one gene that is expressed in multiple ways, or expressed variably is a better way to think about it. So in sickle cell, you may have low conditions if you have one bad copy of that hemoglobin gene, or if every copy of your gene for hemoglobin is wrong, then you're going to have a high susceptibility to all of your red blood cells being impacted by sickle cell. For Fragile X, we can actually look at this protein expression. Remember, genes produce proteins for us eventually, okay, or code for proteins. We can have absolutely no effect whatsoever. You would never even know you were carrying the disease. It can go all the way up through extreme mental retardation. It's actually one of the highest causes of inherited mental retardation that we see in medicine. It can also lead to balance and tremor disorders, infertility in women. What does it actually look like? So here's the X chromosome. This is in a male. How do I know that? There's an X and a Y chromosome associated with the sex chromosomes. Okay. In this X chromosome, you see this little tail off the end? That little tail is indicative of, on both sides here, is indicative of that Fragile X syndrome.